Hello, everybody. Welcome to Loop and Learn. And today is an exciting event. You've seen me posting about Smart Insulin, and I'll bet you want to know what it is. We all do. So we get to listen to this today, we get to watch it, and we get to ask questions. So it will be visited with Dr. Michael Weiss, who is at Indiana University and also a co-founder of Thermalin Insulin Incorporated. And I'll go through our regular little disclaimer here. The Loop app is a do-it-yourself closed-loop algorithm, and while it may seem obvious, please consult with your healthcare professional regarding your diabetes management. We are not diabetes managers. Please remember that this project is highly experimental and not FDA approved for therapy, and therefore you take full responsibility for building and running the system, and you do so at your own risk. And just nod to yourselves and say, yes, I understand, I agree. Just as a reminder, about 2 months ago, we started a website called Loop and Learn, very original, and this is a hotspot for anything you need to understand in terms of running your loop. It's an adjunct to the loop groups. We have some building information, but it'll take you through how do you start. It's got questions, it's got important information and announcements. Definitely join it, definitely subscribe. That way you'll get newsletters and anything else that's coming on that's interesting and important to anybody looping. So I would like to introduce Dr. Michael A. Weiss. He is PhD, MBA, and MD, and co-founder of Thermalin Incorporated, a privately held biotechnology company. Gosh, with all those degrees, you'd think he was old, but he's young. At least he looks young to me. So in February of 2020, not all terrible things happened in 2020. JDRF International presented Dr. Weiss with the prestigious Gerold and Kayla Grodsky Basic Research Scientist Award for his work on insulin structure and signaling with application to therapeutic insulin analog design. I'm not going to ask you to explain that. He will explain it to you. His laboratory has synthesized numerous insulin analogs and contributed immensely to the world's understanding of the structures of insulin, pro-insulin, rapid-acting analogs, and basic analogs, and the mechanics of the hormones binding to the insulin receptor. In 2010, Dr. Weiss founded Thermalin Diabetes, LLC, which sought to design novel ultra-stable pump-based insulin analogs, and he currently serves as Chief Innovation Officer of its successor entity, Thermalin Incorporated. Dr. Weiss is chair of the Department of Biochemistry and Molecular Biology and a member of the Center for Diabetes and Metabolic Diseases at the Indiana University School of Medicine. He is also a member of the Department of Chemistry at Indiana University in Bloomington and the Department of Biomedical Engineering at Purdue University. And I picked up a quote that he shared, and I think it's really very, very touching. He says, "It's my hope against my self-interest is that fewer and fewer people will eventually need insulin. But in the meantime, we'll be making sure that people who have insulin remain healthy and without such dreaded long-term complications as renal failure, amputations, and blindness." I love the word against his self-interest. It's something we always bounce around, but Mike, thank you so much on your cold afternoon for joining us. And I'm going to turn this over to you. Thank you. And teach us all about Smart Insulin.
Well, thank you, Joanne.
Perfect.
So what I'm going to share with you this evening are 3 protein engineering approaches to optimizing insulin for use in closed loop systems. And we want to make closed loop systems as easy and as inconvenient and as unintrusive as possible, and to optimize the time and range with the least amount of effort and anxiety as possible. And as you know, there is a pandemic of type 2 diabetes, but for unknown reasons, type 1 diabetes is going up all around the world as well. And so we hope in the future there's no need for insulin, as Joanne shared, but in the meantime, we're working as hard as possible to enhance insulin replacement therapy, make it easier, more convenient, and in particular to enable achievement of tight glycemic goals without the threat of hypoglycemia. Oops, why isn't this? Oops, there we go. Okay, so here's the star of the show, which are the three-dimensional structures of the insulin molecule on the left and the insulin receptor on the right in its form free of insulin. The structure of insulin was solved in 1969 by Dorothy Hodgkin and colleagues at the University of Oxford after 25 to 30 years of effort. And the structure of the insulin receptor has taken equally long time and now is benefiting from the revolution in cryo-EM microscopy to directly image the insulin receptor complex. And so we're right in the middle of a revolution in structural biology that's deepening our understanding of how insulin binds and triggers the receptor to communicate a signal across the membrane. Now, one of the fruits of these many decades of work, and it's appropriate to talk about this now, especially now because 2021 is the centennial of the discovery of insulin in Toronto, and I'll get back to that in a few minutes. And there will be all sorts of celebratory symposia around the world this year. Because all of this accumulating knowledge is enabling us to make rational changes in the insulin molecule to meet unmet medical needs, most particularly to supercharge the function and safety of closed loop systems. Okay, so here on the left two columns are current pumps, traditional pumps and stamp pumps. And our first technical goal is to have miniaturized pumps in closed loop systems. So in fact, there's a vanishing little footprint under your clothes. And here is the target on the right, which would be a pump the size of a postage stamp that would come pre-filled and be disposable and still last 7 days despite its small size and be inexpensive. Now, it's impossible now to have a pump that small that would last for 7 days, not because we don't have the microengineering and microfluidics to make pumps, but because we don't have the insulin formulations that would be rapid acting when they were concentrated sufficiently to provide 7 days of insulin within a small pump. And so the question that we asked here was, could we re-engineer the insulin molecule to make it ultra concentrated and still ultra rapid, like the reformulations of Humalog and Novolog, to make the robust algorithms for closed loop systems feasible and yet have a miniaturized pump? And the answer is yes, and I'll show you the data, at least in pigs, tonight. And then the second technical question we asked is, could we make insulin so stable that it would enable the long-term use of implantable intraperitoneal pumps so that one would only need 3 or 4 injections a year in an intraperitoneal reservoir, and this would take advantage of the extensive peritoneal membrane to facilitate hyperrapid absorption of insulin in a closed loop system, further enhancing the robustness of the control algorithm. Now the pumps exist. They were tested in the 1990s, but insulin is not sufficiently stable for weeks at body temperature, and the degradation of the insulin tends to clog the pumps, and the degradation products are pro-inflammatory. And so the question is, could you make an ultra-stable insulin formulation that would last for 4 months or longer in an intraperitoneal environment in a pump? And the answer to that is yes. And then for both of these goals, could one introduce glucose-regulated switches so they'd be smart insulins? And the time in range that was optimized to 70 to 75% by the control algorithm, could that go to 95% because it was a smart insulin that was in the closed loop system? And the answer to that is maybe, and we're very encouraged by the data that we've obtained just in the last 2 months. And I'll share our progress to date there. So this is a collaborative effort between Indiana University and Thermalin and Case Western Reserve University, where I used to chair the Department of Biochemistry. And it was from Case that we spun out Thermalin. I'd like to thank my colleagues at Thermalin, the CEO, Rick Berenson, the Chief Scientific Officer, Dodd Michael, and also special thanks to an old friend and colleague retired from Eli Lilly, Bruce Frank, who was the co-inventor of Humalog in his career at Lilly, but has been acting as one of our vice presidents for insulin product development at Thermalin. And so I mentioned we had 2 goals. The first with the miniaturized pumps, that's 1A, and then the smart version is 1B, and the second are the ultra-stable insulin analogs for the intraperitoneal pumps, which we hope also could be made smart. But actually, our longer-term ambition is to combine these different goals all into the same molecule so that we would have the holy grail of insulins that would have all these desirable properties in a single molecule. And we start with Banting, Best, Collip, and McLeod, this team of 4. And the personalities and the infighting among this team are legendary in the field, but actually each was a terrific contributor, and the success in Toronto required all 4 of these men to work together. So this is the centennial of the discovery of insulin. It's also the 50th anniversary of the first high-resolution crystal structure of the insulin molecule at Oxford. So it's quite a landmark. Insulin, therefore, is one of the oldest proteins known to science. And it's extraordinary to me that even after 100 years, there's so many important things that await discovery. And it raises the question, if there's still so much to unpeel the onion of insulin layer by layer, that surely that's true of all the other proteins that we read about in relation to cancer and heart disease and other aspects of medicine. So insulin has really been a teacher for all areas of molecular medicine. And in the sociology of public support for biomedical research, really it was the drama of the events in Toronto in the early 1920s that galvanized broad public support for investing in biomedical research. The other star of the show, in addition to the molecules, is the beta cell. And the beta cell is intrinsically smart in the sense that it could measure the ambient glucose concentration and decide just how much insulin to secrete into the portal circulation to go to the liver first. And it's very interesting that Don Steiner discovered that in the secretory granules of the beta cell, insulin is stored as zinc insulin crystals, just as in many historic insulin formulations. And that is for the same reason. The instability of insulin in the beta cell would otherwise degrade and form amyloid and injure the beta cells. And so evolution invented this zinc insulin crystallization, just as Novo Nordisk and Eli Lilly invented zinc insulin crystallization in the 1930s. And here is a picture of Don Steiner in the years after he discovered pro-insulin at the University of Chicago. And I was very privileged in my career to be one of Don's younger colleagues. And his legacy and memory inspire all of this work. And here is a picture of Don's office. And this was taken in the weeks after Don died. And on his table always was this plastic model of the insulin molecule in a plexiglass box. And when the university wanted to clean out the office, no one knew where this model came from or whether it was important. And so my colleagues at Chicago contacted me and to do some detective work. And it turned out that this model was made by Dorothy Hodgkin, and she's shown at the top right around 1971 when the structure was being determined. And it was hand carried to Chicago as a gift to Don Steiner from Dorothy by one of Dorothy's students, Tom Blundell, who went on to become dean of the college at Cambridge University in the United Kingdom. And so now this model, instead of being thrown away, is in the Museum of the University of Chicago because of its great historical importance in the century of studies of insulin around the world. And so here on the right, we see the Hodgkin structure. It was the fourth crystal structure of a protein ever to be determined. And it was the first that was truly beautiful. This is the zinc insulin hexamer with its striking six-fold symmetry. And Tom Blundell, who I mentioned as the mailman for the model, was the first author of the Nature paper in 1971. He had studied architecture before doing crystallography, and he likened the beauty of the insulin structure to the rose window at Notre Dame, shown on the left. And this, you can see the similarity. And I'd like to thank Pierre Demetz, who's now retired from Novo Nordisk, for sharing with me this slide. Now, unfortunately, this beautiful structure proved to be a failure in providing guidance for design of insulin drugs that would emulate the surface of this. And now we know why. This is the inactive storage form of insulin that protects it from degrading in the secretory granule of the beta cells. When insulin is secreted, the central zinc ions, shown here in yellow, pop out because there's very little zinc in the portal circulation. And then these hexamers disassemble to form dimers and monomers. It's the monomer that binds to the insulin receptor in the liver and around the body to communicate the insulin signal across the cell membrane. Now, the structure of insulin when it binds to the receptor was elusive for many decades. And I was privileged to be part of an international team that included Don Steiner and also one of Dorothy Hodgkin's first graduate students, the late Guy Dodson at the University of York in England, and Colin Ward, who's no longer with us in Melbourne, Australia, and Mike Lawrence, who's my colleague and collaborator now in Melbourne, Australia. He's on summer holiday right now, and it's like 100 degrees in South Australia, but we Zoom regularly. And the crystal structure and now EM structures of the hormone receptor complex revealed that the B chain of insulin partially unfolds to detach between domains of the receptor. And this happens in such a way as to bring the two subunits of the dimeric receptor together, and that in turn causes the signal to propagate across the membrane to enable the receptor to phosphorylate its cytoplasmic tails and then initiate a whole cascade of events in cells to initiate all the complex events that insulin regulates. And this is important for not only diabetes, but metabolism in general, development, and in cancer. So goal one, which we call device-driven engineering, was to make analogs that could be 5 to 10-fold more concentrated than normal insulin solutions, the U100 solutions. So these would be U500 or U1000. It would be ultra rapid acting like FIASP, that is better for pumps and closed loop systems than Humalog or Novolog. And it would be ultra stable in order to make possible pre-filled disposable pumps. And this sounds like a wish list that is impossible, which is perhaps why we had this opportunity to solve it, because easy things had already been accomplished in the very gifted teams at Novo and Lilly and Merck and so forth. And so our inspiration in the lab as we teased each other was from the movie Mary Poppins. And you may remember Jane and Michael Banks fantasizing about the perfect nanny. And they said, would play games, all sorts, and there were 10 other qualities of their dreamed of nanny. And then sure enough, Mary Poppins came down with an umbrella from the clouds. And so that was our wish list for insulin. And we're systematically checking off these qualities en route to what we hope will be the holy grail insulin that combines them all in a single molecule. And so the fundamental problem for miniaturizing pumps for closed loop systems is that insulin is a very sticky protein. And so if you take wild type human insulin or even insulin Lispro or Aspart, and you go from U100 to U500, then you get a molecule which changes from an ultra rapid insulin to something that's like NPH microcrystalline suspensions. And this is true in humans, and it's also true in pigs. And so to validate pigs as our working model, here's a comparison of FIASP from Novo versus Lilly's U500 in blue. And you can see how much slower the onset is of insulin action with U500, and then it keeps going after FIASP stopping working after 240 minutes to 360 minutes. So that's why you just can't concentrate insulin to make smaller and smaller pumps. And so this challenge motivated us to try to optimize insulins. And we took advantage of fundamental biophysical principles that pertain to proteins in general, such as alpha helical propensity, reverse hydrophobic effect, exposed nonpolar surfaces, and the isoelectric point, to try to get an insulin that would be a monomer or dimer without zinc, but nothing bigger, not forming hexamers, and not requiring zinc to be stable in a formulation with a shelf life of at least 30 days, as with current Humalog, Novolog, FIASP formulations. And so here's the fruit of many years of getting there. This molecule at U500 is shown in blue in the pigs, and the FIASP from Novo is in red, and they're statistically indistinguishable, the same onset and the same offset. So this in principle could go to clinical trials now without a closed loop system, because there are a growing number of patients with type 2 diabetes and extreme insulin resistance requiring 200 to 400 units of insulin per day, for which there are no good insulin products. Lilly U500 is used off-label in insulin pumps in some of these patients. Their hemoglobin A1Cs go down. Typically, you find patients in this group with A1Cs of 10%, and they go down to 9%. But that's still very high and associated, as you know, with a high risk of microvascular complications, and probably macrovascular complications as well. So this ultra-rapid, ultra-concentrated insulin in pens would, I would imagine, be of immediate impact in these vulnerable populations, which disproportionately involved patients in Appalachia and underrepresented minorities in American cities and indigenous American Indians. But it really would be of broad impact to enable this dramatic miniaturization of pre-filled disposable pumps, which I think would be transformative, at least for adolescents with type 1 diabetes. One of the features of adolescence is resistance to parental controls and sense of immortality and so forth. And for whatever reason, the T1D exchange has found that in the United States, that the average A1Cs in the 16 to 24-year-old age group is about 9.5%. Due to adherence issues and lifestyle choices. And the same is true in Western Europe. And so our hope is that the successful clinical introduction of a miniaturized, inobtrusive, disposable, easy to use pump would encourage better adherence in adolescents. And there wouldn't be this decade of very poor glycemic control with its associated lifelong risk of microvascular complications. So it turned out that this molecule was more clever than we were. We tried to match the stability of Humalog, which is the red bar here, and this is a time in hours in an accelerated degradation assay where we're reading formation of fibrillation or frosting of the vial. And the bottom blue bar is the U500 ultra rapid formulation that we have as a candidate for clinical testing. We're very pleased that a major pharma has recently agreed to work with us on clinical trials of this. And you can see that the blue arrow at the bottom, it's still going at 180 hours when we stopped the experiment. So this turned out to be much more stable than we had designed, which is all to the good. And so here on the left is the famous picture of the first insulin pump, and then in the middle is a current pump, not the patch pump, but a current external pump. And then just to say that here's our engineering goal is to have a stamp size pump. And here is an illustration of such a pump, which would also be very flat, so its profile under the close would be very low. It would also have Bluetooth and communicate to a continuous glucose monitor and to a control algorithm, last 7 days, perhaps more, and also no manual filling. It's pre-filled, and that would be very helpful for any patients that have visual impairment. And the insulin would be stable for up to 6 months on the shelf at room temperature. Now, here's where we dream of doing everything better. So we'd like to have this ultra-rapid U500 also be smart. That is, to have a glucose-regulated conformational switch so that the control algorithm protecting patients from hypoglycemia would be amplified in its effectiveness by the intrinsic glucose responsive nature of the hormone. And that would mean that in simulations that even without a bihormonal pump that would be injecting rescue glucagon, that the system itself could be made more robust than closed loop systems. And the closed loop systems now are actually terrific and getting better every year. And so just imagine if the closed loop system were using a smart insulin. So that pushed us to continue trying to introduce glucose responsive switches. And we were very lucky to get support from the JDRF, the NIH, and the Helmsley Charitable Trust. As an aside, the JDRF and Helmsley grant programs are now paused for COVID. And we're grateful to NIDDK for keeping us going during this difficult time. And so here we come back to the structural switch that happens in all insulins as it binds to and triggers the receptor. And the question we posed for itself was, how could we exploit this endogenous mechanism to create a smart insulin? And the answer is we're not there yet, but I think we're very close. And I'll show you the latest data over the past few months and why we're so excited. So here was the idea. So when the B chain of insulin opens to bind to the receptor, this part here, the C-terminal beta strand, which we depict as an arrow, detaches from this part here, which is the alpha helical domain of insulin. The arrow then inserts between the two subunits of the insulin receptor to initiate the signal propagation. And so in our imagining, we wondered what would happen if we put a glucose binding element at the end of the A chain, at the beginning of the A chain, and an internal mimic of glucose at the end of the B chain to make this closed. The closed form of insulin is very stable, but it's without biological activity. And so we imagined that at high insulin concentrations, we could have exogenous insulin come in and compete with the internal mimic to enable the hormone to open and therefore bind to the receptor and be active. So that I can't show you tonight because we are still optimizing glucose binding elements. But we had a good fructose binding element. Fructose is an isomer of glucose. They're both monosaccharides. So for the principle of this, we turned from a glucose binding element to a fructose binding element to create a model fructose responsive insulin as an engineering test of this whole scheme. And that's worked beautifully, and I'll show you some data. And so here we were very lucky that a prominent diabetologist at UCSD, Dr. Alberto Hayek, and the Diabetes Research Connection in La Jolla provided support to a younger colleague at Indiana, Dr. Yen-Shen Chen, shown here. And Yen-Shen took this scheme and modeled in the cryo-EM derived structures of the insulin receptor hormone complex. And here, the blue blobs are actually the modification sites. And he did this with the help of Dr. Mike Lawrence, my colleague in Melbourne, who's now on summer holiday. And they created a molecule based on Lispro or Humalog that put an internal mimic of glucose or fructose here with an aromatic diol. And they put a known fructose sensor here at the beginning of the A chain. And here at the bottom is the longer B chain. So this is a molecule that's modified at A1 and at the lysine of Lispro. It also contains, at this red asterisk, a favorable amino acid substitution from avian or bird insulins, and that enhances the binding of insulin when it's open to compensate for any reductions in activity due to the two modifications. So that's a detail. And here are the key data that Yen-Shen obtained. And each of these panels represents some kind of receptor activation or post-receptor signaling. And in each case, he's testing these liver-derived cells, either in 50 millimolar fructose or 0 fructose. And at the far left, in each case, it says KP, that's insulin Lispro, the active component of Humalog. And the big bar here and the gray bar here indicate that Humalog doesn't care about fructose. Well, we know that. Humalog is not a glucose-dependent hormone. It's not a fructose-dependent hormone. It's just a good insulin. And this particular panel is looking at activation of the insulin receptor. And the red bar is the fructose responsive insulin, and the red bar is big in the presence of fructose, and it's small in the absence of fructose. So we have a fructose-dependent hormone. If one removes the internal aromatic diol mimic of glucose, that's the middle bar here, and it's high with fructose, and it's high without fructose because there's no switch. So that's a control. And these three panels represent downstream signaling events. So this, for example, is the AKT kinase, which is so important for communicating the signal to GLUT4 to go to the surface of muscle or fat cells. And in liver cells is important for the control of gluconeogenesis. And this is the transcription factor, FOXO1, that controls the enzymes that undertake gluconeogenesis. And this is GSK3, another downstream target. And in each case, we have fructose-dependent signaling of this design scheme that Yan-Chen is testing with fructose. What I'm delighted to share is in the last few weeks, we've had some success in converting this design from fructose to glucose. And so the prospects look very good over the next year for having the world's smallest glucose responsive insulin. And by what I mean by world's smallest is that a lot of the smart technologies for insulin involve elaborate materials engineering, so that there are depots of insulin and polymers under the skin, and these complexes change in their physical properties at lower high glucose to enable the insulin to get trapped or to be liberated from these subcutaneous polymers and go into the bloodstream. So there's been more than 30 years work on engineering glucose responsive polymers and matrices for this purpose, and they're supramolecular. But this is just the insulin molecule with two small tags on it. And so even when it circulates in the bloodstream, it could reversibly go back between the open conformation and the closed conformation as a switch that's operative and reversible both in the subcutaneous depot and in the bloodstream. Okay, so that's where we are with, that's called a unimolecular glucose responsive insulin. And then how do we make things like ultra stable so that they could be for many months in the reservoir of an intraperitoneal pump? And our approach here is to put a engineered peptide tether between the A chain and the B chain, where the connecting peptide of pro-insulin is normally there. And that's a 35 amino acid connector, which is very loose and floppy. And it's so long that insulin can still rearrange its shape to form amyloid as a pathway of physical degradation in the pumps. Now we know that these termini have to separate for the B chain beta strand to insert between the two receptor subunits. And so there's some play that's needed. And we've defined an optimal length of a foreshortened connecting domain or C domain to bridge the A chain and the B chain. And that indeed confers even more stability. We had some of these insulin solutions with this bridge going for almost 2 years at 37 degrees, shaking in glass vials in the presence of an air-liquid interface. And these are all terrible things for insulin. And yet it stayed active. And so we believe that this class of insulin analogs would be appropriate to be stable for many months and just a few refills per year in an intraperitoneal pump. And so we've been testing these, maybe I should call these hyper stable insulins, by doing a very simple kind of experiment that is gentle agitation in glass vials with this air-liquid interface at some elevated temperature, 37 degrees, 45 degrees, 60 degrees. We've even boiled some of these solutions and then injected them into diabetic rats to see if they're still active or not. And current products undergo within a few weeks at 37 degrees complete inactivation due to formation of these ropes of insulin fibrils, these inactive aggregates. This is an electron micrograph showing what happens to wild-type human insulin under these conditions. So I'd like to show you a single rat experiment that's representative of this heat agitation test. It was conducted by my case colleague, Dr. Faramars Ismail Beghi. And you may know his name because he was the principal investigator of the famous ACCORD trial in type 2 diabetes, which showed that if you have long-standing type 2 diabetes, then glycemic targets have to be moderated, and that the more aggressive A1C targets are associated with more mortality. In any case, Dr. Ismail Beghi is an MD PhD who has 2 careers treating patients at the VA hospital and university hospitals in Cleveland, and also having a rat lab. And we've been privileged to collaborate with him for almost 20 years. And so let me show you these data. So in these X's are what you see for the blood glucose levels if you just inject buffer alone, which is lily diluent. So the rats are not eating at the beginning of this experiment, and the blood glucose concentration goes down a little bit and recovers. This is the glycemia. You can see these are grossly hyperglycemic animals. The mean starting glycemia is around 400 milligrams per deciliter. They don't have functioning beta cells. And then in blue is a Lantus injection. The blood glucose concentration goes down to about 60 at this dose and then gradually comes back up over the 12 hours of the experiment. And then in red is what happens if you do this heat agitation test for 10 days with Lantus. And there's no activity left at all. The Lantus has been completely inactivated. And then in green triangles are one of the single chain insulins that we engineered to be a basal insulin. And in orange is what happens if you agitate it at 37 degrees for 110 days, which is more than 3 months, and there's no change. So it's completely resists any kind of impairment of activity under these extreme conditions for a long time. Now, you wouldn't want to have a basal insulin in a pump, either external or intraperitoneal, but we're getting the same results with rapid-acting, ultra-stable single chain insulin analogs. And so now the experiments that we're doing in the coming few weeks is whether we could put in a glucose responsive switch into these super stable, rapid-acting single chain insulins toward that holy grail idea of having one molecule that has all the Mary Poppins list of desirable qualities like Jane and Michael Banks in their nanny. So fully active after 3 months. Now, as an aside, I mentioned that we're in the middle of a pandemic of type 2 diabetes and also a global rise in incidence of type 1 diabetes. And it was really the unmet needs in the developing world that led me to go into diabetes clinically and as a research career. When I was a medical student, I had an opportunity to go for a month to East Africa. And this was just at the beginning of the detectable rise of the trend toward more diabetes around the world. And in 1980-81 in East Africa, type 1 diabetes was a fatal disease almost always because there was such a problem with the cold chain and getting insulin to East Africa, that if a child or an older patient developed type 1 diabetes, he or she often died within the first year. And the insulin that did get there was usually stored in clay pots that were buried in the ground. And so one of the spin-offs of our work on trying to create insulins for intraperitoneal pumps in closed loop systems is that there could be a humanitarian application of giving the super stable insulins around for free in places like Ethiopia and Kenya, Tanzania. And I've spoken with the Gates Foundation when we started this project. And, if we do have a regulatory path in the West, it's possible that as a spin-off of this effort, there could be a humanitarian arm, as there is with the beautiful work being done by the Gates Foundation in HIV and tuberculosis and other infectious diseases. So I'd like to end by thanking my gifted students at Case Western Reserve who've just graduated, Anisha Reggae and Michael Glidden. They're both now interns in internal medicine. My former students, my fellow Bala Murugan Daladan, who was just promoted to be a research assistant professor, Yanru Yang, who did the three-dimensional structure of pro-insulin, my case colleagues, Nelson Phillips and Fairmars Ishmael Begi, who I mentioned, two retired colleagues who were in the lab for many years in previous decades, Professor Quinchen Hua and Zuli Wan, and then again to thank the National Institutes of Health and the American Diabetes Association, the Helmsley Charitable Trust, the JDRF, and the Diabetes Connection for their generous support over the years. And it has been really a surprising and wonderful thing that these funding agencies would invest in our efforts over many years, and this has been since 1985, to strive to reach nearly impossible goals. And now we see that there's light at the end of the tunnel, and it's an amazing coincidence that as our insulin engineering is coming to fruition, there has been in parallel by our colleagues around the world, parallel advances in better control algorithms and better pumps and better continuous glucose monitors. So I think uniquely over the next 5 years, there's a chance to put all these methodologic and technical advances together to make a closed loop system that is really as close as we can make to a beta cell in the era prior to beta cell stem cell therapies that will be a cure for type 1 diabetes and eliminate the requirement for pumps or insulin formulations. So thank you so much for the chance to spend part of Sunday with you in this ongoing adventure, and I'd be delighted if there are questions.
Oh, there are lots of questions. And there's also a movement underfoot to make you president. I think the thing I just have to say is, at least from my perspective, I just have tears in my eyes, that there's this possibility. It's very, as much as I understand what you spoke about, it's really very, very exciting. So let me get to some of the questions that have been coming in. Well, there was a comment that I thought was just actually totally perfect. So it just simply said, a stamp-sized pump that's in a closed loop and lasts 7 days, and I don't have to pick up insulin at the pharmacy. Okay. So we got people here who are ready to sign in. The first question actually came in a couple of days ago to ask you to talk about the stem pump. So are you able to say anything about a timeline on the stem pump?
So I know that there's a very gifted group of brilliant engineers working on the stamp pump, and I've seen prototype breadboards of different components, but I don't have a background in electronics or materials engineering, so I can't knowledgeably speak about it. But I'm extremely impressed by the team that Rick Barrington has put together to work on the engineering of the stamp pump.
Okay, so I've also recently been reading about physiologic devices, which is an implantable pump. And I remember there was one 25 years ago, or maybe 30 years ago, and it was really large and it had a reservoir. What you're looking at with the stamp pump, which we all at this point want tomorrow, are there other, would you work across any device that will deliver your insulin?
Yes, yes. So for the intraperitoneal pumps, they wouldn't be stamp size because we want a 3-month supply of insulin, but they'd be less than half of the size of the 1990s Medtronic's pump.
Okay. So you're the insulin part.
Yes.
Is someone asked, is it correct to assume that each of these parts of this project would need to go through its own trial process with the FDA?
Yes, and our hope would be that the regulatory path for the insulin analog formulations would be independent of the stamp pump and enable their initial use for a few years in pens.
Okay. Another question, would you consider licensing to existing hardware providers, people who are already making pumps in the system?
Yeah.
Okay. Can you say who they are? I had to ask. Are there contenders that you're looking at, or?
So we've had wonderful conversations with a whole variety of innovative device manufacturers. And we actually have a partnership with one on making better tubings and inserts that go under the skin so that they last longer with less irritation under the skin, which also isn't my skill set, but there's been great teamwork from Thermalin with Dr. and my colleague in Philadelphia, Thomas Jefferson.
Yeah, so it's capillary biomedical people, right?
Yes, capillary biomedical.
Okay. Yeah, we had Mark Estes come speak to us. I've worked with Mark with Asante pumps way back, and we've been tracking them, and several of us have done studies, focus groups with them as well.
So Professor Jeff Josephs.
You betcha.
Okay. Right.
Because that will be an integral part of what you're doing, is the ability to deliver safely over a period of time.
Yes.
Okay.
By the way, I don't think that the present insulin analog formulation would allow mail order delivery in the summer of a pre-filled pump, because what happens if your mailbox went up to 50 degrees centigrade? But it could be without refrigeration at the pharmacy. But our holy grail dream, if we put all these favorable features together, would be to enable mail order delivery even in the summer.
I'll drive anywhere, but many of us would, but I understand what that issue is. A question about what is the lowest amount of delivery? What would that be? That she mentions she has a 4-year-old where they have to dilute a U50 to get to the Omnipod's lowest delivery. Would it ever work with a U500 with young children?
That's a good question. I'd have to explore that. I think we'd be close to the limit, and one might have to dilute the U500 to U250. Okay. Now, the wonderful thing about this analog formulation is that unlike Humalog or Novolog, there's no change in stability with dilution. So when you dilute Humalog, its actual shelf life goes down. But that's not true here, or it has very, very small effects.
Okay. And when you mentioned major pharma companies you work with different than device manufacturers? You're talking about insulin manufacturers?
Yes.
Can we know anybody you're talking to yet? You know, I'm going to ask, but you don't have to answer.
I don't think I'm allowed to say, but
Okay.
the agreement was signed in November.
Okay. That's cool. Then a question about, can you fit enough? Obviously, the answer is yes, but how do you fit enough insulin into such a small form factor?
Because it's so concentrated.
Okay. So
That's the key.
what would it look like? Would it look like a capsule, a medical capsule, that amount of insulin only, or even less?
It would be more than a capsule because we call it a postage stamp, but it's the bigger postage stamps that you would find in stamp collectors' international albums,
Okay.
and not a square. It's round, and it has a little thickness to it, and it gives you enough volume, just enough volume to have a week's worth of insulin, not for type 2 diabetes, but for insulin sensitive type 1 diabetes.
Would this be something that could also go sit in a jacuzzi with us?
We don't know yet because we haven't built the stamp pump, but that's the goal.
Okay. I'm willing to volunteer for that research, by the way.
In fact, you know who someone who sent me a email of encouragement was Gary Hall Jr.
Okay.
And as you know, he developed type 1 diabetes in between Olympics.
Mm-hmm.
And one doctor told him he'd have to stop his competitive athletic career. And his doctor is an endocrinologist in Los Angeles who's been one of our advisors. And she went to the poolside with him to make sure he could train for his next Olympics. And he won another gold medal.
Yeah, you can't stop us diabetics, you know. Question for one of our medical doctors. Hypothetically, what happens if the 3-month supply of insulin inside the intraperitoneal pump leaks or ruptures into the peritoneum?
That would be catastrophic. It hasn't happened in the experience with the Medtronic's pump, happily. It's always been the reverse, that the insulin can't get out. And you probably know that in Montpellier, France, there's a continuing ongoing use of the Medtronic's intraperitoneal pump with a few dozen American patients that are required to fly back to the south of France every few months to have their pumps cleaned. Which actually sounds very nice. I went to visit Montpellier, and it's quite a beautiful place to visit. But no, it's an inpatient or in-hospital procedure to flush out the reservoirs with a pH 10 or 11 solution to dissolve the amyloid deposits.
Wow. There was a Dr. Salam who started the initial internal pump. Does that follow on from his research? I'm
not sure. I met a different doctor there who was my age, named Gerold, I think.
Okay.
He's Mr. Internal Pump now. He trained in Boston too for a few years, and so we overlapped in Boston.
Okay. I think all of us are just sitting back trying to catch our breaths at what we're hearing. If you were to give a time frame, is there anything you can say? I grew up hearing there'll be a cure in 5 years. I mean, we all heard that. But this seems actually doable. You actually understand the structures and the mechanisms that are working. Do you have a sense of time frame?
Well, I wish it were yesterday.
Thank you.
In fact, so some of the people on the call may remember the famous JDRF conference in 1990 in Monaco, with the Prince of Monaco saying that the 1990s would be the decade of the cure.
Mm-hmm.
In 1990, I was much younger, and I thought 10 years was an infinitely long time. but my sense now is that there's a well-defined regulatory path for insulin analog formulations at the FDA. It's not tricky, and that phase 1 clinical trials can be done relatively inexpensively and quickly with a few dozen patients. And that efficacy information is obtained even in phase 1 because the toxicity is hypoglycemia and the efficacy is control of glycemia.
Mhm.
And my looking into the regulatory timeline suggests that within 5 years, one can go from the analog to the product. And Lilly did this, going from the discovery of Bliss Pro to its clinical introduction in 1998 in a 5-year time.
Very exciting. My goodness. There are questions, but it's more enthusiasm. There's a tremendous amount of appreciation for what you're doing and your very serious focus on this. So thank you for all the years you've been involved with this. I just see someone posting something that his father worked at the University of Buffalo to develop the pacemaker. Are there lessons to be learned from the process they went through in the 50s to develop an internal device?
Boy, I'm sure there are. I'd love to know more about it. Since my mother was born and raised in Buffalo, and my father went to medical school in Buffalo, I probably should know more about this story than I do.
I will connect you. David, I'll give you the link up. I just want to say thank you. If there are follow-on questions, may I just shoot them over to you and post them with our group?
Oh, please do.
And if there's any like breaking news, please feel free to let us know. And if you need volunteers, I bet this group would put a hand or two up. And if there are trials that we might be able to participate in, I'm sure we'd be interested. So please keep us in mind.
Oh, Joanne, thank you so much. It definitely will be needing participants in the upcoming clinical trials, which we hope will happen.
We're excited. Mike, thank you so much. Please stay safe and warm in the cold weather, and we'll be in touch. Thank you. Stay safe.
Good night, everyone.
Good night.